introduction. I'll share my screen and try to find my talk. Okay, so can you see that okay? And can you see my pointer?
Yes, we can see it.
Okay, so the title, as it says, is about how cerebral blood flow is regulated, both normally and during a range of pathological diseases. So I'm going to try to persuade you that these little red cells here, which are contractile pericytes located on capillaries throughout the CNS, but also throughout the body, these play a major role in controlling the energy supply to the brain. As an overview, I'm going to tell you about the regulation of energy supply in the brain. Can I just ask, can you see this floating panel at the top or not? Can you see the full top of the screen?
We can see the full screen.
Okay, that's fine.
It says, yeah, overview.
Yeah, we can see the overview, yes.
How is energy supply regulated? I'm going to show you that pericytes can constrict and dilate capillaries, which has been a controversial subject. And I'll then show you that astrocytes regulate capillary diameter, whereas neurons control arterioles. Having dealt with physiology, I'll move on to pathology and look at the role of pericytes in the ischemic pathology of the brain when you have a stroke. I'll show you they have a key role in the onset of Alzheimer's disease, and I'll look at their role in COVID-19 as well. And throughout all of this, I'll try to consider some therapeutic approaches. So let's start out with energy supply regulation in the brain. This is Homer. As you know, his brain is about 2% of his body's mass, focused on the things that everybody's brain is focused on. But it uses about 20% of his resting energy supply. And so that's why we have this dense network of blood vessels throughout the brain, which seem to leave very little room for any neurons. They bring in oxygen and glucose in the blood, and that's used to power your thoughts. For those of you who are familiar with hippocampal slices, these are the neurons, of course, these little bobbly things, but you may be less aware that these cells need a vast network of blood vessels to power their actions. And even with the energy arriving in all these blood vessels, you can't have them all operating at a maximum firing rate all the time. If some neurons up here want to fire, for example, then they have to release local vasodilators to bring in, to make the blood vessels larger and bring in more energy supply. So this is the topic I'm going to cover today, at least in the physiology section. This is a simplified diagram of the plumbing of the brain. How is blood flow controlled? Well, blood comes down from the surface of the brain through the cortex, through penetrating arterioles, which are ringed by arteriolar smooth muscle cells. And until recently, it was thought that adjusting the tone of these smooth muscle cells was what adjusted blood flow as it goes into the capillaries, where nutrient exchange occurs with the neurons. But it turns out it's much more complicated than that, because even back in the 1870s, it was known there are little cells with processes around capillaries, which look as though they may be able to contract and relax to adjust blood flow at the capillary level. And that's what I'm going to be focusing on today. So these were discovered by Ebert in Germany and Rouget in France. And I want to have a moment's digression on the rise of studies on pericytes. So they were discovered back in the 1870s. They were named 50 years later, but really nothing happened for 100 years. But now there's an exponential rise in the number of papers being published on pericytes, with more than one paper being published every day, but they remain seriously understudied. If we look in the brain, for example, they're present at 50 to 150 microns along capillaries. They're a major cell population, but if you go to PubMed, you'll find that only one CNS paper in 1,000 mentions pericytes. It's even worse in the heart. In the heart, pericytes are the second most numerous cell type after endothelial cells. The myocytes come in a poor third, but only one cardiac paper out of every 5,000 in PubMed is about pericytes. So this suggests that we're missing out on therapeutic opportunities because pericytes contribute to all diseases involving ischemia, a decrease of blood flow. So that includes stroke, heart attack, spinal cord injury. And I'll also show you they have a key role in Alzheimer's and probably in COVID-19, diabetes and cancer. But they're largely ignored by scientists and big pharma. So I'm a kind of missionary for pericytes. You should go out and study them. I want to show you they can constrict and dilate capillaries. Now, it turns out that in 1920, a Nobel Prize was actually awarded to Augustus Krogh, a Danish physiologist, for showing that there were contractile cells on capillaries. He studied them in skeletal muscle, and this is a little sketch in his Nobel lecture, showing that depending on the muscle, the work being done by the muscle, you could have roughly half the capillaries dilated and providing providing energy to the muscle, and half of them constricted and not providing much blood flow. And valuable work has also been done since by Don Puro's group on retinal pericytes. But it turns out that much of this original work of Krogh has simply been forgotten. And so I want to start out by showing you that pericytes really can constrict and dilate blood vessels. So this is a blood vessel in the retina where it's easy to get a large piece of capillary in focus at the same time. This patch pipette is being used to depolarize a pericyte underneath here. If I play the movie, you'll see that when it's depolarized, it squeezes off the capillary completely, showing the power of these cells, at least to shut down blood flow. Now, this movie, if I play it one more time, shows another remarkable thing, which is that pericytes talk to each other. There's a pericyte here and another one down here. You can see that after this one constricts, when we depolarize it, this one constricts here. This is done by an electrical signal going along the endothelium outside, which forms the wall of the capillary. So if you plot the diameter of the capillaries at this central stimulated pericyte and at the ones on each side, you can see they repeatedly reduce their diameter, whereas in between on the capillaries, there's no diameter change. So pericytes are the only contractile cells on capillaries. Now, it turns out that neurotransmitters also regulate capillary diameter, and I want to show you this in human tissue. We get human tissue from neurosurgical operations. If you have to remove a glioma from the brain, often you need to remove some normal tissue above the glioma in order to access it. And then we can get tissue that looks like this. If you do histology, these are pericytes with a bump on a log morphology. What we've labeled here is the basement membrane in green using this lectin. You can see the pericyte sitting here. And I'll show you their processes in a minute. Here's one in a live cortical slice. So we get this tissue by waiting outside the operating theatre, getting the tissue when it's removed, and then cycling back to the lab and making normal brain slices from it. These are red blood cells inside this human capillary. If you apply noradrenaline, a transmitter which is normally released from the locus coeruleus in the brain, you see that it squeezes the capillary near the cell body here. And that's shown in this diameter plot at the bottom. If you then apply glutamate, it dilates it, and you can then wash that out again. So pericytes constrict capillaries near their stomata. Neurotransmitters adjust the diameter. Why is it near the soma of the pericyte? Well, if you take mice in which the pericytes are labeled fluorescently, you can see these fluorescently labeled processes going along the capillary and also around the capillary. And it's only the circumferential processes which can adjust the diameter of the capillary. So if we count the number of those processes, you find that nearly 80% of them are within 10 microns of the soma. And that means that if you apply a constricting drug in this brain slice, then you get a big constriction near the soma, but only a small constriction further away. And indeed, if you plot the diameter as a function of distance from the soma, before you apply the constrictor, you find the diameter is actually slightly larger near the soma. We believe the soma induces growth of the endothelial tube. But when you apply the constrictor, then you get a preferential constriction at the soma. And I'd like you to try to remember this graph here, because we're going to be coming back to it later when we turn to looking at Alzheimer's disease. Now, the ability of pericytes to constrict capillaries matters because it turns out that in the brain, it's the capillary bed which has got most of the resistance to the blood supply within the brain itself. So 4/7 of the total resistance is in the capillaries. 1/7 is in the arterioles, which are also have an adjustable diameter. So 4/5 of the total adjustable resistance is in the capillaries. Okay, so we want to know what happens in vivo. And to do this, we've used 2-photon imaging to look through a hole in the head above the somatosensory cortex of a mouse, in which the pericytes are labeled red and the green is painted green by injecting Vikcy dextran. And this vessel on the right is a penetrating arteriole going down into the brain. And this is the first order capillary and then 2 second order capillaries coming off it. And we can image this and measure the diameter. And if I show you, well, the result is shown in this movie at the bottom, when we stimulate the whisker pad of the animal to put input into this region of the brain, the capillaries dilate first and only later the arterioles dilate. I'll show you this up here. So when we stimulate the whisker pad, the capillaries dilate, but only later the arterioles dilate. I'll play that one more time. The capillaries dilate and only later the arterioles dilate. And that's important because it shows you that this capillary dilation is an active relaxation. It's not just that they get pushed open because the arterioles have dilated and raised the local blood pressure. So they dilate faster than arterioles. Normally it's about 1 second faster than rather than 3 seconds. And it's not just faster, they also dilate more than the capillaries dilate on average. So they show a lot of potential for contributing significantly to the control of blood flow physiologically. In fact, if you know about functional imaging signals, bold fMRI signals of the brain, which are widely used by psychologists to investigate brain function, it's likely that the onset of those signals is dominated by the fall of resistance as the pericytes dilate. So when you look at those little red blobs in bold functional imaging pictures, you should be aware that what you're looking at is the increase of blood flow produced rapidly by pericytes dilating. So that's the background. Pericytes have the potential to control blood flow. I now want to show you there's a kind of spatial segregation in how this control of blood flow occurs. How do we want to control blood flow? Let's suppose we're designing the energy supply to the brain, we're evolution. It turns out that if you do an energy budget of the brain, most of the energy use in the brain occurs at synapses. So more than about 60% of brain energy use, what you had for your lunch, is consumed on pumping out ions which enter at synapses, mostly postsynaptically, but also presynaptically and in glial cells. So it's appropriate that it's synapses which release messengers to dilate blood vessels. And a lot of this is driven by the release of neurotransmitter glutamate. Glutamate is the transmitter at 85% of the synapses in your brain. Of course, it opens rapidly acting AMPA channels and slower acting NMDA channels. It also raises calcium in astrocytes, and calcium goes up in neurons because of glutamate activating the NMDA channels. And these rises of calcium can activate enzymes like phospholipase D2, which makes arachidonic acid, and nitric oxide synthase, which makes nitric oxide. And these substances generate vasodilators, prostaglandins from the arachidonic acid, or nitric oxide from the nitric oxide synthase, which can act on pericytes or on smooth muscle cells. And this is done in the context of a background constriction produced by noradrenaline released from locus coeruleus axons. So we wanted to know, is the pericyte control of blood flow produced, how does it occur? One way we've looked at this is to patch clamp the pericytes. This is a genetically labeled pericyte, which we've then patched with a different dye in the patch pipette so we can make sure we've patched the right cell. If we then apply glutamate to evoke neuronal activity, we get an outward membrane current. This is very different to what you would see if you applied glutamate to a neuron, where you would get an inward current as you open sodium passing channels. What's happening here is a potassium current is being activated. I'll explain how in a minute. And if you stimulate neurons, this is actually in the cerebellum, stimulating the parallel fibers and looking at the membrane current in a cerebellar pericyte, you also get an outward membrane current. This is blocked by TTX, and the size of the current is enough to generate a roughly 10 millivolt hyperpolarization. Now, because the resting potential of these cells is about minus 40 millivolts, and that means that they have voltage-gated calcium channels, L-type voltage-gated calcium channels, tonically activated, this hyperpolarization will close those calcium channels, and you'd expect that to decrease calcium influx and relax the smooth muscle within the myofilaments within the pericytes. And indeed, if you image pericytes within the brain, like I just showed you, and stimulate the synaptic input to this area and use a calcium dye in the cells to measure the calcium concentration in their processes, you find that pericytes on first, second, and third order capillary branches all show a fall of calcium concentration when you start to stimulate the neurons. And this is presumably generated by these potassium channels opening and hyperpolarizing the pericytes. As a result, you can see the pericytes dilate the capillaries. This is an experiment done in a brain slice where we stimulate axons going across the cortex, and we image the capillaries and measure their diameters. Now, to do these experiments in a brain slice, because we've cut off the locus coeruleus axons, which go to the which produce basal noradrenaline release and basal tone of the pericytes, we have to pre-constrict them. And we either do that by superfusing noradrenaline, or in this case, a thromboxane analog, which reduces the diameter of the capillaries, as you can see here, when you apply it. We then stimulate and get a dilation of the capillaries. Okay, it's only a 20% dilation, but it occurs. And we can then do some pharmacology. This is blocked by TTX, as you'd expect, because we hope it's action potential driven. It's blocked by the AMPA receptor blocker, NBQX, but surprisingly, it's not blocked by the NMDA receptor blocker, AP5. And I'll come back to this later. So how is this happening? We wondered whether astrocytes are involved, because astrocytes seem well designed to control blood flow at the capillary level. They have end feet which go onto capillaries, but they also have other processes which sense what is going on in synapses. And so to investigate this, we patch clamp astrocytes with a calcium buffer in the patch pipette. Now, this might be a high concentration of a fast-acting buffer like Bacta, or it might be a low concentration of a slow-acting buffer like EGTA. And when we go whole cell, this will go into the cell, and if it's a fast, strong concentration, fast-acting buffer of a high concentration, it will buffer calcium transients and stop these messengers being generated. And so that you might hope that would stop the dilation being produced. This shows the experiment being done in a brain slice. There's some capillaries here. This astrocyte has got an input onto this capillary just here. And if we patch onto that astrocyte with a low concentration of a slowly acting calcium buffer, then we pre-constrict the capillaries with this thromboxane analog. We go whole cell and put this very feeble buffer into the cell. And when we stimulate neuronal activity, we get a dilation of the capillary. If we then do it instead using a high concentration of a fast calcium buffer, now in this very carefully chosen example, when we stimulate neuronal activity, we get absolutely no dilation at all. Buffering the calcium has stopped the dilation. And on average, we get about a 70% reduction of the dilation occurring. To sum up a load of boring pharmacology, we find the way this works is that neuronal activity triggers or activates AMPA receptors, which lead to the release of ATP from postsynaptic neurons. That acts on P2X receptors on astrocytes, raising the calcium within the astrocytes. That then activates a phospholipase, which ultimately generates arachidonic acid, which generates prostaglandins, which cross the membrane and act on pericytes by binding to EP4 receptors. So that's this pathway summarized here. Neurons are active, they release glutamate onto other neurons, ATP is released, that makes calcium go up in astrocytes, and prostaglandins are released onto pericytes. However, it turns out there's a totally independent pathway by which neurons release glutamate onto interneurons that activates a calcium influx through NMDA receptors. Remember, NMDA receptors were not involved in this pathway, and neither was nitric oxide synthase, but there's a more classic pathway by which nitric oxide synthase is activated, releasing nitric oxide, and that leads to a dilation of arterioles. So there are these two pathways for physiological control of blood flow. So that was physiology. I want the rest of the lecture to be devoted to three types of pathology: stroke, Alzheimer's disease, and COVID-19. I'm going to start with stroke. 1 in 5 women in the audience is going to have a stroke, and 1 in 6 men is going to have a stroke. Men have less strokes because they die earlier doing other silly things, riding their motorbikes too fast, that kind of thing, smoking. Now, a classic experiment on the causes of some neuronal damage in stroke was done by Ames back in the 1960s. He did a very simple thing. He took some ink of the sort you might have written a letter with back then, and he injected into the blood of a rabbit. And in control conditions, he showed that the ink goes through all the blood vessels of the rabbit. But if you do middle cerebral artery occlusion to mimic a stroke, so you squeeze the middle cerebral artery or occlude it, so the blood flow is cut off for an hour, and then you open it again and let the blood flow come back for an hour, he found that even after waiting, injecting ink into the rabbit after that simulated stroke and recovery, so it simulates treating someone for their stroke by removing a clot, nevertheless, the black stuff doesn't go into the microvasculature. If you look at a cross-section of a brain, you see there are these white areas where the brain no longer has a functioning blood supply. And this is called the no reflow phenomenon. And we investigated this in brain slices, looking at capillaries, the microvasculature. We found that if we applied ischemic solution, which stops the production of ATP by glycolysis and mitochondria, then you evoke a constriction of the capillaries where there are pericytes. And this isn't surprising. If this is a pericyte with its arm around a capillary, then normally you have lots of ATP-driven ion pumping, which is keeping the calcium concentration low. Then when you have your stroke, the ATP level falls, so ion pumping stops, and the calcium concentration will rise. That will then activate the myofilaments in the pericyte and squash the capillary. This seems simple. And this also occurs after you do middle cerebral artery occlusion in vivo. That was a retina. This is now in vivo in the cortex. What we've done here is, this is in a mouse where the pericytes are red. You may just be able to see them. There's green stuff in the blood, in Vikcy albumin in gelatin, and there's vessel labeling with this purple isolectin here. And what we've done is to do 90 minutes of simulated stroke and then reperfusion for 70 minutes. And we then inject this green stuff in gelatin and cool the animal down so the gelatin sets. And we can see which vessels were perfused with blood and which ones were not. You can see that although the blood comes back through many vessels, it doesn't come back through quite a substantial proportion of them. These vessels are labeled, but they have no green blood in them. And you can measure the diameter at many millions of places on these vessels using a semi-automated procedure called a PhD student. And if you do that, what you find is that the diameter goes down specifically near pericytes, or at least more near pericytes than not near pericytes, both in the core of the lesion and in the penumbra. So pericytes are constricting vessels here, and that is reducing the blood flow. And indeed, if you look at the blood flow through the brain during the middle cerebral artery occlusion, the blood flow goes away. When you open up flow again, it comes back. There's some reactive hyperemia, but then it ends up being only about 60% of the original value. And so the question is, why is this fall of blood flow so long lasting? Excuse me. And we answer this question by imaging brain slices. If you image a brain slice capillary for a long time in normal solution, its diameter is constant. Whereas if you apply ischemic solution, then the diameter very rapidly decreases, as you can see here, just as we showed in vivo. But then if you apply propidium iodide, which normally can't cross the membrane because it's got 3+ charge, but if the membrane of the pericyte is ruptured, then it can go in and label the genetic material. It fluoresces red. And we found that after just an hour of ischemia, the cell membrane permeability is broken down. So the cell has constricted, it's squeezed the capillary, but it's then died while still squeezing. And once it's died, its ATP level is low. And if you remember your muscle physiology lectures, you need ATP to relax muscle, not to contract it. So it's squeezed the capillary, it's made it be a small diameter, but it's going to stay that small diameter now because the cell is dead until a microglial cell comes along and removes that dead cell. So here's the graph of the time course of the constriction and of the death of pericytes, which occur roughly after 15 and 45 minutes with this profound ischemic insult. This is actually a stronger insult than if you have a stroke, because we're stopping all ATP production. And this occurs in the white matter as well. And so we think that after the capillaries are constricted, the rise of calcium triggers the death of the cell. We know that if we take the calcium away, we can reduce the amount of death occurring. So this long-lasting constriction seems to be likely to be the reason why the pericytes can't relax and the blood flow stays low. This decrease of vessel diameter doesn't just decrease the blood flow, it can stop it flowing altogether. This is the entire vascular bed of a bit of brain with the arteriole here and a venule over here. You can see in places, here we've colored the blood red, there are places where the blood flow suddenly stops right by pericytes, which are colored green here. And in some cases, there's a neutrophil sitting at that block. And we think neutrophils have got stuck at the narrowing of the blood vessel which has been produced there, because the distance from the neutrophils to the pericytes is very low compared to the distance between the pericytes. So this long-lasting decrease of blood flow, we think, reflects the constriction and then the death of pericytes, which means that pericytes are a therapeutic target in stroke. If you have a calcium channel blocker present while you're doing this whole train of events, it slows the constriction of the pericytes because the calcium won't rise so much. It also reduces the amount of pericyte death occurring. Now we're using oxygen glucose deprivation. So the cells die during the OGD. They die even more afterwards, but you can prevent all that by having nimodipine present. If we now mimic a clinical situation by only giving the nimodipine when we reperfuse, you can see that the blood flow now, instead of ending up less than it was before, comes back to be more than it was before, and that improves the behavioral outcome on a scale called Neuroscore. So giving nimodipine on reperfusion after MCAO increases blood flow and improves recovery. So that was stroke. I want to turn to Alzheimer's disease now. You're probably aware of Alzheimer's disease. As you go from being a healthy control through mild cognitive impairment to late onset AD, a whole load of things change. You start to deposit amyloid beta plaques with some kind of time course like this. Tau gets hyperphosphorylated, synapses are lost. But the very first thing that happens is there's a decrease of cerebral blood flow. And this brown curve plots the decrease in blood flow due to the disease with the change that normally occurs due to aging removed. And this is a big change of blood flow. It goes down about 45% in the parts of the brain which are affected. Now you might ask whether a 45% reduction matters, but it does. If 100% is normal blood flow and normal function, then if you go down to by just 20%, you suffer a loss of ability to maintain attention. This may be due to adenosine being released in the brain and then suppressing glutamate release from synapses. By the time you get down to the range of reduction occurring in AD, you suffer damage to myelinated axons, spatial memory deficits, inflammation is triggered, and then you get synapse loss and neuronal damage. And over this whole range, protein synthesis is being inhibited. So that's going to have long-term effects. I'm going to have a digression for the role of microglia in triggering these events. You may have seen movies like this. This is a 30-minute loop in which microglia move their little processes around, looking for bad things happening in the brain, to phagocytose, for example. If you binarize these images and then add them all together, you can see that over an hour or two, the microglia gradually survey the entire brain. Now, as they do that, they interact with all the cell types, astrocytes, neurons, just here, and also blood vessels, capillaries. Now, why might this matter? Well, as I said earlier, most of the vascular resistance within the brain is in capillaries. Microglia synthesize vasoactive agents. For example, they synthesize thromboxane A2, which is a constrictor, endothelin-1, a very powerful constrictor, and they can also make prostaglandins, which are dilators. So given that, and it's known that microglia extend processes towards blood vessels in stroke, might they do that in Alzheimer's disease? It turns out they do, at least in Alzheimer's disease mice. When we measure the distance between pericytes and microglia, that's really reduced in the AD mice. So could that be having an effect on the properties of vessels? We think that it is, and we wanted to look at what's happening to pericytes early in AD. And again, we wanted to do this in humans. So it turns out that in the hospital at Queen Square, near where I work, if you're dementing for no known reason, they will happily remove a biopsy of your right prefrontal cortex to look at if there's any obvious cause that's treatable. And when they do that, they find that roughly half the patients have no amyloid beta deposition. They're not getting Alzheimer's, whereas the other half are depositing amyloid beta and are getting Alzheimer's. We could then access that archive tissue and look at the pericytes on it and see how the diameter of the capillaries is affected. And when we do that, we found a mean 8% reduction of blood vessel diameter. Doesn't sound like very much. You have to measure about 9,000 diameters to pick it up. But it turns out this is produced by pericytes, because if we measure the diameter as a function of distance from the pericyte, we find that in the patients who aren't getting Alzheimer's disease, the diameter is larger near the pericyte. And like I showed you in brain slices, without a constrictor present. Whereas the patients with Alzheimer's show a selective constriction at the pericyte soma. And this difference in profile predicts a 50% reduction in cerebral blood flow. I'll remind you, this looks very similar to what I showed you in brain slices, applying a chemical vasoconstrictor. So given that we think this is being produced by the disease, we can then look as a function of the amount of A beta being deposited. And what we find is that if you look at the amount of A beta being deposited here, as soon as it starts being deposited, there's a very rapid reduction in this extrapolated diameter at the soma. So this occurs early in the disease, and it's probably contributing to the onset of the disease, as I'll explain. We also looked in transgenic mice. They also show a reduction of diameter that's larger near the soma of pericytes, and also shows this classic variation with distance from the pericyte soma. Interestingly, this doesn't happen in the cerebellum, which doesn't show any amyloid beta plaques, and there's no change of diameter of the arterioles or venules. So the reduction of blood flow is produced specifically at the capillary level. And these constrictions may trap neutrophils, as I mentioned earlier. So what's the mechanism? We went back to our human brain slices, and we made oligomers of amyloid beta, which are thought to be the toxic species. And when we apply them to these brain slices, they evoke a slow decrease of diameter. Now, of course, this is an hour-long experiment, whereas AD develops over decades. But nevertheless, this seems to be a potential explanation for what we see in the AD patients. And what's more, the EC50 for this effect is similar to the level of soluble A beta found in the brains of patients with Alzheimer's disease. So what we think is happening, I can't show you all the data, but what we think is happening is that amyloid beta generates reactive oxygen species, which release endothelin-1, a powerful vasoconstrictor, that acts on endothelin receptors to dump calcium from internal stores, which then activates the myofilaments to constrict capillaries. We could try preventing this by blocking this ROS-producing enzyme, blocking endothelin A receptors, although it's known in the literature that blocking established constrictions is difficult for endothelin. We also tried another approach using C-type natriuretic peptide, which activates guanylate cyclase, and that stops calcium being dumped from stores. And also, by a Viagra kind of mechanism, it raises the concentration of cyclic AMP, and that turns off the myofilaments. So to test these ideas for how we might prevent this constriction happening, we evoke a constriction with amyloid beta. If we block the activating pathway with these drugs, you halt the constriction in its tracks, but it doesn't reverse very rapidly. But if we give the CNP, then we get a rapid reversal. So we're now working on ways of translating these into drugs which might reverse the decrease of blood flow which occurs early in AD. So I've shown you A beta evokes pericyte constriction, which decreases blood flow, and suggested ways of preventing this. Now, this reconciles genetic evidence for the importance of A beta with the fact that the earliest event in AD is vascular dysfunction. Interestingly, when energy levels fall, that upregulates the enzyme which makes A beta. So there's a positive feedback loop here, which will increase the amount of A beta, produce the amount of tau hyperphosphorylation and downstream synapse loss. But it also suggests how some patients develop AD. If you are a professional footballer and head the ball a lot, or a boxer, you suffer transient reductions of cerebral blood flow. You suffer energy supply loss also if you have sleep apnea and don't breathe properly at night. All of these patients tend to get AD, and that may be because this upregulates the enzyme which makes A beta and sets off this positive feedback loop. A key question is whether to what extent synapse loss is due to this pathway up here, and which is promoted by the energy falling, and to what extent it's a direct result of the loss of energy supply. And here's a review, you can read more about this in. So in the last roughly 10 minutes, I want to turn to a more popular disease and the role of pericytes. I guess you've heard of this. This is the COVID-19, the SARS-CoV-2 virus. It binds to its cells by the spike protein on the outside of its envelope. And it turns out, by chance, and this is how I got into this, that the receptor for the spike protein is an enzyme called ACE2, angiotensin converting enzyme 2, which is called a receptor in the context of its virus action. And this is located moderately specifically on pericytes, also on some endothelial cells. So both in the brain and the heart, it's mostly on pericytes. Now, COVID-19 is unusual in that although you inhale it up your nose and it goes into the lungs, so it's primarily in a respiratory disease, it actually affects tissues all over the body, or it can do. There's a fall of brain blood flow. There's a fall of blood flow in some other organs as well, and you get damage to many organs. How does this happen? The effects on other organs may be via release from the lung of pro-inflammatory cytokines, but another possibility is the virus itself can spread through the blood. This does occur with high viral loads. And it can then access ACE2 in other organs by crossing endothelial cells either via transcytosis, entering the brain via the olfactory pathway. It can hijack a ride on macrophages going into tissues. Brain pericytes are known to be infected. Now, to explain what goes on, I need to remind you of the angiotensin, the renin-angiotensin system. You may be familiar with this in the kidney, where if you suffer a loss of body fluids, renin, an enzyme, is secreted into the blood from the kidney. And that converts this precursor, angiotensinogen, in the blood into a 10 amino acid peptide, angiotensin 1, which is then converted by angiotensin converting enzyme, a target of blood pressure drugs, into angiotensin 2. And this acts mostly on angiotensin 1 receptors to produce a constriction and increase your blood pressure again. There's some opposition to this by angiotensin 2 receptors. And 20 years ago, that was the whole story. But now it's known that these peptides can be converted by angiotensin converting enzyme 2 into another peptide, which can act on the so-called MAS receptor to cause dilation. So when the virus binds, it is thought, it doesn't occlude the binding site for these drugs, but it internalizes and removes the enzyme from the membrane. So you no longer make this angiotensin 1 to 7. To study this, you can't use rats or mice because the sequence of their ACE2 has changed from the human version, in particular at this K353 position. However, hamsters haven't got this mutation, and they're a good model for looking at the properties of COVID-19. So we use golden hamsters to do these experiments. If you do immunohistochemistry and label for ACE2, you find that it's mostly in cells which co-label for these other pericyte markers here, NG2 and PDGFR beta. And if you quantify where the label is, nearly 80% of it is on capillaries rather than arterioles, and most of it is on these PDGFR beta positive pericytes. So the action is on capillary pericytes. Now, what happens if you apply angiotensin 2 to pericytes in the brain? You get a small constriction, about 8%. This is blocked by the AT1 receptor blocker. So I told you this is the main mode of action of the angiotensin 2. If you, however, block either angiotensin 2 receptors or, more importantly, MAS receptors, you get a much bigger constriction produced. So this tells you that activation of MAS receptors is opposing the constriction produced by angiotensin 2. So how is all of this affected by the virus? To look at this, we haven't used the virus itself, although we're starting to use pseudovirus now. Instead, we just use the receptor binding domain of the virus, which is what binds to ACE2. If you just apply the RBD on its own, it doesn't do very much. There's a small constriction produced. But if you look at the response to angiotensin and then apply the receptor binding domain, it massively potentiates the response to angiotensin. This is specific to angiotensin. It doesn't potentiate the response to another vasoconstrictor, this thromboxane A2 analog. And to make sure this is being produced by the virus itself, binding to ACE2, we use a mutated version of the receptor binding domain with just one amino acid changed, and we find that then abolishes this potentiation of the constriction. And this gives you a 12% reduction of the constriction. So normally angiotensin 2 acts on angiotensin 1 receptors to constrict, but it also generates angiotensin 1 to 7, which opposes that constriction. When you have an ACE2 bound to COVID-19 situation, that removes the ACE2, and now you don't have activation of MAS. So you might predict that if you block ACE2, that should have the same effect, and indeed it does. When you apply angiotensin 2, you get this potentiated constriction in the presence of the RBD. You get the same potentiated constriction if you block ACE2, and this potentiated constriction can be blocked by the AT1 receptor blocker losartan. And so it's interesting, there are currently some clinical trials going on looking at whether losartan can reduce some of the effects of COVID-19 on the brain and other organs. Because if there's a long-lasting decrease of blood flow in the brain, that could contribute to some aspects of long COVID, for example. So to conclude this section, the virus removes ACE2 and deprives cells of MAS activation. AT1 blockers might be useful, but don't get too excited. This needs extending to in vivo data using pseudovirus. It's unclear exactly how large an effect on blood flow would be produced. It's unclear what the real angiotensin level in the brain is. By the way, there's an endogenous angiotensin system in the brain. It doesn't need to come from the kidney, but where it does come from is a key question. So in today's lecture, what I've done is I've shown you how energy supply is regulated in the brain at both the capillary and the arterial level. I've shown you pericytes regulated at the capillary level, and neurons, and this mechanism goes via astrocytes, whereas neurons control arterial regulation of blood flow. In stroke, pericytes play an important role by constricting blood vessels and then dying in rigor, giving you a long-lasting decrease of blood flow. Pericytes, we believe, play a key role in the initiation of Alzheimer's disease and offer therapeutic targets. Pericytes probably have some kind of role in COVID-19. And in the future, I think studies are going to focus on the diversity of pericytes, and they will bring in more diseases, including diabetes. Pericytes are important in the pancreas, things that happen in the retina, the brain, and the periphery in all of these diseases, and also glioma and epilepsy. These are the people who helped me do the work, and I'd be happy to take questions. Thank you for listening.
Thank you very much for this very interesting talk, Professor Attwell. We can already start with the Q&A. We have plenty of questions already. Is there any evidence of pericyte involvement in neuropsychiatric disorders like schizophrenia?
I would say there's no direct evidence. There are other neurological conditions where there's a fall of brain blood flow, and that may reflect reactive oxygen species generation and endothelin release, as we're suggesting occurs in Alzheimer's disease, for example. Synuclein-related disorders which generate reactive oxygen species may decrease blood flow in that way, and that could contribute to the dementia which can be associated with Parkinson's. But I'm not convinced about schizophrenia yet.
Yes, thank you.
We have a quite similar question. Has pericyte dysfunction been implicated in any other neurodegenerative diseases besides Alzheimer's disease?
There's a good example of it in spinal cord injury from David Bennett's group in Canada. It's published in, I think, Nature Medicine, where he showed that trace amine release after spinal cord injury leads to pericytes constricting and reducing the blood flow, and hence to spinal cord degeneration.
Thank you. And we have another one from the audience. You showed impressive evidence for pericytes being involved in diverse fatal pathologies involving insufficient blood flow. To what I understood, one of the issues during migraine headache is a change of cerebral blood flow. Sounds to me like pericytes are likely to play a role here as well.
I think it's quite likely. Martin Lauritsen's group in Copenhagen has shown that the spreading depression, which is associated with migraine, is in part produced by 20 heat release. Excuse me. And we know, I'm losing my voice. We know that that constricts pericytes. Let me cough for a second.
Yes.
Right.
Thank you. Thank you very much.
Yeah, so we have quite a lot from the audience. Another one, the minimum, sorry? Oh. The minimum diameter of the capillary seems to be notably smaller than the size of the erythrocytes. Is it possible that in some circumstances, the local influx of the oxygen through the capillary is completely blocked, but the outflow of the CO2 dissolved in plasma remains partially preserved? Can that cause pH regulation problems?
It's an interesting question. We're interested in how red blood cells squeeze through the vessels. There is a cell-free layer at the edge of the vessel, so it's possible, I guess. There would still be some oxygen delivery in that plasma layer as well.
Another one, amyloid beta has been shown to have antimicrobial effects, suggesting a potential role in innate immunity. Could the constricting effect of amyloid beta has on pericytes have a protective effect against infection? For example, if there is a systemic infection, amyloid beta forms brain microvessels, constrict, and there is less flow of microbe-infected blood into the brain.
Okay, it's an interesting hypothesis. I wouldn't want to sign up to that one. It seems a bad idea to shut down your brain blood flow under most circumstances, actually.
Yeah. Thank you.
We have one more from the audience. Most of Alzheimer's disease is sporadic. Do you or other studies observe the dysfunction of pericyte in sporadic AD models, for example, APOE4 variants?
Sure. The patient data that I showed, my voice has come back, phew. The patient data that I showed was from sporadic AD patients. So yes, we do. APOE4 is known, at least in mice, to decrease cerebral blood flow. And we're interested in whether that's a pericyte action.
Okay. I don't see another question from the audience. Oh, wait. No. But I do have a quite general question.
Okay.
You mentioned it was discovered that pericytes regulate capillaries over 100 years ago. And after hearing this talk, I'm kind of wondering how or why were pericytes neglected so much? Like, was it just too difficult to study them, or was there some other reason? Or
I think it's a question of what's fashionable, actually. I think for a long time, let's say in the 50s and 60s, most cerebral blood flow people were more interested in the big vessels and how pressure varied through them. It was measurable, unlike the pressure in the microvasculature. There's much more interest in the microvasculature now. And it's become more appreciated that most of the brain resistance is actually in the microvasculature. Of course, that doesn't mean it's all variable. It's mostly the pericytes on the first few branch orders of the capillary network, which can constrict and dilate. But nevertheless, especially in pathology, they have the power to turn off the hose completely, as it were, to turn off the blood flow completely.
okay, thank you.
Thank you. Maybe I can ask one last question. Do you think there is any difference between the different COVID strains with regard to their effect on pericytes?
Well, they bind, it's all going to be determined by how they bind to ACE2, probably. And so the different strains do bind to ACE2 differently. In fact, some of the more modern, if I can call it that, variants do bind to rat and mouse. When we started, this wasn't the case, but now the modern variants will bind to the ACE2 in rats and mice. So you can use those variants to study them in rats and mice now, which is a big technical advantage. So I would assume that the differences will largely be through binding to ACE2, but there's also this protein cleavage step in the internalization of the virus, which may also differ depending on the mutations which have occurred in the virus.
Okay, thank you very much for this very interesting talk again.
You're welcome.
And we will have a short break before we welcome our next and last speaker.
Okay.
So see you everyone in 5 minutes at 5:15.
Thank you.
Thank
Thanks.
you.